Welcome to the second keynote address of this year's RSS conference. It's a very great pleasure to be able to introduce Anuj Srivastava. Anuj is a professor of statistics in the Department of Statistics at Florida State University. He's been there since 1996, shortly after he finished his PhD at the Washington University in St. Louis. He's also a regular visitor to Europe. He's held visiting positions at INRIA and Lille in France, and also in Durham. Anuj has very broad research interests. He in statistical computer vision, in functional data analysis, and in shape analysis, some of which he will, of course, tell us about today. He has a very distinguished research track record, over 250 peer-reviewed papers and several significant books, including a Springer text in functional data analysis. So with no further ado, I'll let Anuj talk, but I will say one thing from a procedural point of view. The Q&A for this session will be done via the conference app, so please open up the conference app and be prepared to put in questions in there, and we'll deal with the Q&A in that way. But before the Q&A, Anuj, the floor is yours.
Thank you. Thank you very much for the introduction. I want to extend my thanks to the organizers for giving me this opportunity to discuss my research. This is a sort of a mixture of different topics in this talk. It's not about getting deep into technical issues on any particular depth, but trying to be more of a bird's eye view of a certain field. Let me make sure this works. It does. Okay. So I want to talk about complex data analysis. As statistics is getting more modern, we are getting data which is more complex, which is big, large, which is structured. And so we have to come up with techniques for handling this kind of a data. And this talk is somewhat in that direction, motivating this kind of research for statisticians. The subtitle of this talk is Kendall meets Fisher-Rao. And I want to make a connection of this sort of modern data analysis with some very old results in statistics, which perhaps are somewhat forgotten by now, but they are helping build more modern foundations. All right, so this is a very, very brief overview, is to say how seemingly distant results from very different parts of statistics come together to build this field, to help found this field of structured data analysis. It's also interesting to see how knowledge, skills, and traditions got handed down academic inheritances from advisors to students. So on a historic note, I make notes of those ideas. Okay, so I want to start with what is structured data analysis. Perhaps this is a new phrase to some of you. Perhaps some of you are very familiar with it. So structured data analysis is about quantifying statistical variability in objects. Could be natural objects, could be man-made objects from biology, bioinformatics, chemistry, anatomy, and so on and so forth. And so how to mathematically represent such complex objects, how to capture statistical variability, and how to do inferences and testing and other things that we are comfortable doing in statistics. I just want to give you some examples to ground this discussion. So for example, a lot of us are interested in representing and capturing anatomical variability, maybe subcortical structures in a human brain, and associating the structures, the morphologies of these brain parts to some kind of diseases, perhaps cognitive diseases, ADHD, PTSD, and so on and so forth. Another example from the field of bioinformatics is protein structure analysis. Maybe some of you heard about this, a giant breakthrough that this Google's DeepMind team had on estimating the structures of protein from the data. And once you have estimated these structures, then somebody has to do the analysis of those structures. The key thing in protein structure analysis is somehow how to relate the structure of a protein to the functionality of the protein. What role does it play? And it's very important in things like drug discoveries and so on. Another example I'll mention is biological growth. There are many different kinds of biological growth where you observe shapes over time, and you want to be able to write down some kind of statistical models and be able to quantify what does it mean to be normal growth, what does it mean to be abnormal growth, and how to quantify, how to be able to make decisions about those things. And finally, I'll mention this very common, very popular discussion right now on COVID data. So here are some examples of COVID curves. These are from a few different countries in Europe. And so we get this data, different curves for different cities or different states or different countries. And the question is, what are the common patterns that are occurring? What is commonality among these different curves that we are observing? So all these are examples, few examples, there are many, many more of structured data analysis. And people study these things using different kinds of tools. You may have heard of topological data analysis, where one focuses on topological variations. You may have heard of geometrical data analysis, which is what I'm going to talk about, is focusing on the geometries of the objects that we are interested in. Okay. But the discussion starts way back. It goes back to the work of Professor Fisher-Rao. In 1945, he wrote a paper, or a series of papers, which were remarkable in many different ways, one of which was his contribution to this thing called Rao distance. At that time, it was called a Rao distance. It was basically, if you are given a family of probability density functions, let me see if I can point this out. Yeah. If you are given a family of probability density functions, you pull out two elements of this density, how to quantify differences between the two probability PDFs within that family. And he did this using a certain metric, which was eventually called, later on called Fisher-Rao metric, because it also occurred, this discussion also occurred in papers of Fisher in 1920s. So this Fisher-Rao metric takes this form. This is like an inner product. And in our introductory inference classes, we also call it the Fisher information matrix. And this matrix is quite useful in different parts of estimation theory. And this Fisher-Rao metric for comparing PDFs has played roles in many, many different domains, including this famous field called information geometry, where Amari and his colleagues have made good use of this. One important use of Fisher-Rao metric, or the Fisher information matrix, was to lower bound the variance, the error, expected error, in any estimator, any unbiased estimator, and Fisher-Rao, and in parallel, Harold Kremer proved that the estimator is lower bounded. The error in an estimator is lower bounded by the inverse of the Fisher information matrix. Okay. So this was an important result, and it's helped establish maximum likelihood criteria as a prominent approach for doing estimation, because if you have such an estimator MLE, it actually achieves the lower bound. You can't do any better than that. Separately, completely in an independent way, Anil Bhattacharya wrote a paper in 1946 for comparing PDFs, but this time not restricting to parametric families, but any two PDFs. And he wrote this formula where you take the square roots of the PDFs, do this integral, and put a cosine inverse. And this was called a Bhattacharya distance. Okay? And so I think at that stage, he even called it just a divergence. He didn't quite make the connection with other things going on at that time. But it turns out at the root of this Bhattacharya distance is a certain kind of inner product. And this inner product is given by this equation on the top. And nowadays, it's called the non-parametric version of the Fisher-Rao metric. And you can see the parallel. This is the non-parametric Fisher-Rao inner product, and this is the parametric Fisher-Rao inner product, which I flashed earlier. And you can see there the parallels. The variation in this case are due to derivatives, because this is restricted to a parametric family. But here, the variations, the inner products of these perturbations are unrestricted, because you're non-parametric. Any function is allowed to change the PDF. And so this formed the non-parametric setup for Fisher-Rao metric. And the solutions were much simpler. In fact, he even wrote down the distance between any two densities under this non-parametric Fisher-Rao. It was as simple as this. What is surprising is that this, despite its simplicity, didn't gain traction. I mean, in our classes, we talk about Fisher information matrix, but we seldom talk about the non-parametric Fisher-Rao metric, which turns out to be a much simpler solution and in many ways. Okay. So this original formulation was designed for some PDFs, but not general functions. We are interested sometimes in functional data analysis. So it turns out there are different versions of this parametric Fisher-Rao. This was the version for PDF I flashed a few seconds ago. But if you're interested in more general functions, it takes a slightly different forms. So for example, if you think about cumulative distribution function, you can go from CDF to PDF by taking a derivative. So similarly, in the same way, you can replace the terms for the non-parametric Fisher-Rao for PDFs by the corresponding derivatives of the functions or CDFs. Okay? So it's not important to try to remember these formulas, no big deal. The implications are much more interesting and tremendous. One of the implications, which I don't know whether it was realized that early in the history, was this metric is invariant to a certain transformation. That if you take these three functions, v1, v2, and g, and you compose all three functions by a same gamma. Gamma is a certain class of function. I'll come back to that in a minute. But if you take these functions, this inner product remains unchanged. So in mathematical terms, this non-parametric Fisher-Rao metric has a wonderful property that is invariant to transformations of the time coordinate axis. This gamma is somehow transforming the time, and it remains invariant to that. Now, changing topics, going to a slightly different field in structured data analysis, looking at David Kendall's work. He was a pioneer in the field of shape analysis, how to define shapes, how to mathematically represent and make it precise so that we can actually do statistical analysis. So his work on shape, he defined that shape is a property that if we rotate, translate, and scale objects, then this property remains unchanged. He says that's what we are interested in. Okay. This approach, objects were represented by a number of points. They are called landmarks, and I'll show it by example. So suppose we have these two curves, and we want to compare their shapes. In his approach, in Kendall's approach, we will represent these shapes with some number of points, these blue dots. They are called landmarks. It has to be same number of landmarks across two objects, and we have to know which landmark is registered or matched with which landmark in the other object. Once we have that, then we do certain transformations because these transformations don't change the shape. They are in some sense nuisance. We want to get rid of them. So you center each object, you scale each object, and then finally you rotationally align object. So you may take the first object, this set of points, call them P, this set of points, call them Q, G is a rotation. You search over all possible rotations of the second object that it best aligns with the first object. And once you have done this, then you have gotten rid of all these transformations, and then you can somehow interpolate between these two shapes. You can calculate a distance and quantify how different they are. Okay. So that was his approach, and there is a mathematical version of it. There's no need absolutely to go understand all this notation. I just want to mention some of the topics because I want to come back to these. Mathematically speaking, each shape, each planar shape was represented by k landmarks in R2, and we want to remove some of the groups by centering and scaling. Rotation is not so easily removable, so the way we remove rotation is we form these equivalence classes where we say all possible rotations of the object have exactly the same shape, so that's an equivalence relation. And when we talk about shapes, we don't compare individual shapes, we compare these equivalence classes. Okay? And we do so by doing this minimization. These vertical bars, they represent the Euclidean norm. And important question was, why choose this Euclidean norm? Of course, that's a very obvious thing. We are all trained to use Euclidean norm by default. But it works for this interesting, important reason that if you take two objects, in this case, I just take 2 points, P and Q, if I rotate both the points by exactly the same rotation, the distance between them doesn't change. Okay? It's obvious, it's so obvious we don't even think about it. We simply use this result day in, day out. That if you have two objects, the Euclidean distance between doesn't change if you rotate them by the same rotation. This, in fact, turned out to be a fundamental reason why you could fix one object and just find the best rotation of the second object. All right, so that's that. There has been tremendous amount of work done in this field. In fact, the field of statistical shape analysis has been developed by several pioneers, a couple of students of David Kendall and Professor Mardia, who is in this audience, and Professor Mardia's student, Ian Wright. And so they have lots of papers, in fact, a couple of fantastic textbooks that are widely recognized in this field and used for this. So they developed intuitions into what is the shape space of these objects, how to model these shapes using probability distribution, how to do analysis, how to do testing. So they've developed a very rich field of statistical tools based on the ideas of David Kendall. The limitation of this approach is the obvious one that I've already highlighted, is that objects are represented by landmarks. So if you are given contours, if you are given more real data, real objects, how to put landmarks on them. And you have to put not only the landmarks, you have to put the same number of landmarks, and you have to register them across. And so this registration problem, that which landmark here matches with which landmark here, is a tremendous problem. It's the gist of the whole problem in shape analysis, according to me. And so it seems to me, just looking at this picture, it may be a better idea to even work with just continuous curves as it is, rather than trying to discretize the problem. Okay? The question is how to do that. And so Harold Kremer, who I mentioned earlier, had a student, remarkable career, Ulf Grenander, who did a lot of different works and in fact inherited Harold Kremer's chair as in the Mathematical Statistics Institute at Uppsala. And Grenander, with one of his, at that time, younger colleague, Michael Miller, developed this field of computational anatomy. It's a slightly different approach to studying shapes and studying structure. In this approach, you don't put landmarks on the object, you try to deform the whole object as is. So the idea is you try to deform, so you get images, these are the slices. This is somewhat old work, so you used to work with 2D slices, now one works with 3-dimensional objects. And so one looks at these objects and you try to deform one object into another, and you try to compare or quantify the differences with how much deformation you had to do to take that one object into another. These deformations are also groups, but they are much higher dimensional, in fact, potentially infinite dimensional groups. But there is a very rich theory, there is a whole field of computational anatomy where people use this kind of approach. The last topic I want to mention in terms of sort of collecting the tools for what we want to do is the field of functional data analysis. And I noticed there are several papers in this conference from this field of functional data. In functional data analysis, the observations are full functions or curves themselves. And what one does is, this is a work of many, many people, including the famous book of Silverman and Ramsay. They impose a certain metric structure on the function space, and you exploit that matrix structure to compare functions, to average functions, to put probability distributions on functions. Most notably, people take some sort of orthonormal basis and project infinite dimensional space into some truncated finite series and go back to sort of multivariate statistical analysis that we are comfortable with. The question is, what is a common metric structure? And a common metric structure is the standard extension of Euclidean norm, which is the L2 norm for functions. I will mention it in the next slide. So this is the inner product between any two functions. The norm between any two functions is given by this. This is the L2 norm. And this is very prevalent. People use it without even thinking about it. I just sat through a paper this morning on in this field, and the whole paper was presented with assuming this metric, but without mentioning it, that there is an underlying metric that is being used here. How good is this metric for functional data analysis? That's an important question. I'll give you this sort of artificial example. In this example, I have lots of functions, and it may not be so clear from this picture, but each one of them is a bimodal function. Each function has 2 peaks. If I use this prevalent technique to average these functions, the average of these bimodal functions is this function with 4 or 5 modes. And that's the fallacy, that's the weakness of using this metric, is you lose the structure in the original data when we start doing statistical analysis. Here is another real example. These are the hospitalization data curves for 25 countries in Europe. And for each country, there were 2 or 3 prominent peaks, maybe sometimes even 4. But when you start looking for a common pattern, you say, let me just average the given data to look for a common pattern, you get these 3 very shallow peaks. The peaks start smoothing out when you start averaging under this approach. So that's a limitation of this approach. Just to drive home the point a little bit further, suppose I have these two functions. They are both similar shape in some sense. They have a peak and a valley, but the peaks and valleys are not aligned. They are at certain spacing. If I use the current approach to sort of compare or interpolate between these two functions, these interpolations look like these dotted lines, which do not retain the structure. Instead, if I do something different, if I register or if I match peak with peak, valley with valley, and the intermediate points accordingly, and then I interpolate between the curves, you see you have a better retention of the structure. And that's what is central to this structured complex data analysis, is you have to do the registration in such a way that when you do statistics, when you start talking about statistical summary, you preserve the structures that you are looking for, that you are interested in the first place. You don't lose them by the simple tools that you have utilized. So there is this notion of registration, which point in this object should match with which point in the other object. Sorry. Now this problem of registration gets more and more complex as the objects get more and more complex. So we're talking about simple scalar curves, but we can have curves in n-dimensional space, we can have surfaces, three-dimensional objects, we can have tree-like objects, we can have networks, we can have graphs. So as things get more and more complex and we want to start comparing them and start doing statistical analysis, the registration problem becomes more and more difficult. In fact, in my opinion, as I said earlier, this is one of the biggest problems in complex functional data analysis, is how to register the objects. Okay. So I want to get back to solving this problem, but with a simple example. Of course, we can do more complex later on. So the question is, if I have these two curves, and I want to figure out which point on the blue curve matches with which point on the red curve, the way I'm going to do it is I'm going to compose this blue curve with a time warping. Essentially, I'm going to move the peaks and valleys horizontally in the blue curve in such a way that the peaks line up with peaks and valley lines up with valley. So I want to transform this blue curve into this green curve so they look well matched in some sense, in this geometrical sense. Okay. So the goal is to find this gamma function which will enable me to do this. And so the question is, how should I find this automatically? Of course, I don't want to sit there and do it by hand. Okay, that will defeat the purpose. So one thought process, as usual, looking at functional data analysis, is to perhaps try to minimize the L2 norm between the function f1 and f2 composed with gamma, and try to find the best gamma. This is in the spirit of Kendall, where we fix one thing and we try to best align the other thing. Okay. It turns out this optimization is degenerate. I can take completely different function and I can make the difference between them arbitrarily close to 0 without even looking at what they are. So this is a degenerate process. What do people do? People recognize that very early. What do people do? You put penalty. When in doubt, put penalties. And so there was that solution, and I will mention something about it doesn't work well, but there was really a mathematically fundamental problem. The fundamental problem was this, that if I take two functions and time warp, this gamma I call time warping, if I time warp by the same gamma, the norm, the L2 norm is not preserved. Compare this with Kendall's analysis of aligning by rotations. In Kendall's analysis, the rotations work because rotations don't destroy the distance between points. Time warping destroys the L2 distance between the point. So using L2 norm for functional data analysis is highly questionable if you are interested in geometric properties of the function. Okay. On the other hand, this is the most prevalent norm. That's the most prevalent framework used in all of functional data analysis today. Okay. What should be used instead is a metric which has a nice property. And I mentioned very early in my talk that non-parametric Fisher-Rao metric has this invariance property. Don't know whether it was recognized that early in the game, but this metric happens to have just that wonderful property we need for functional data analysis. So what we do here is, I'll skip this slide. So what we do here is try to bring these two pieces that I built together. Firstly, I want to use the non-parametric Fisher-Rao metric, which has the nice invariant property, and Kendall's thinking of aligning the objects through transformations to do the comparisons. And this is where I said the Fisher-Rao meets David Kendall. Okay. Although I'm told that, I tried to look for a picture of two of them together, and I couldn't figure it. Nobody agreed that they were together pictured on this at any given time. That would have been just so good. I tried to do Chat GPT, but just to make up a picture someday. So this is the new setup, putting these pieces together, is I'm going to focus on the space of some continuous function. In the spirit of Kendall, Kendall's work was removing rotations. In my work, we are going to think about removing the time warping, using the time warping for aligning the peaks and values of the function. And instead of using the L2 norm or its penalized versions, we are going to directly use Fisher-Rao metric without any penalties, without any choice of parameters, and so on. All that is good so far, mathematically speaking. From a practical point of view, there is still a problem that the Fisher-Rao metric is non-trivial. I mean, if you look at this metric, if it was a standard inner product or a standard norm, we know how to compare and minimize standard norms. This is somewhat non-trivial and requires thinking. So the solution to handle the complexity of the Fisher-Rao metric came from some kind of a square root function. This was derived with my colleagues, Eric Klaas and Ian German. Ian German is in the audience today. A lot of these brilliant guys I worked with. And one solution was that instead of working with the functions directly, do some kind of a square root transformation. You take the derivative, you take the square root, but you preserve the sign of the derivative. So it's sort of like a signed square root. And remember, this has some connection with the square root that Anil Bhattacharya took in 1947. He took the square roots of PDFs to derive a very simple form without making the connection with the metric. What is beautiful about this representation is that the Fisher-Rao distance, which was seemingly complicated earlier on, if I take two functions, form their SRVFs, we call them SRVFs, the L2 norm between the SRVFs equals the Fisher-Rao distance. So something which is very daunting, mathematically speaking, becomes as simple as just L2 norm once you go into this transformed space. And you can do all the analysis in the world that you have ever learned with using L2 analysis in this space and bring the results back. In fact, up to a translation, this is a bijection. You know, one can even write down very detailed mathematical results. And so this leads to this Kendall-type alignment that I'm going to take a function, fix its SRVF, and I'll try all possible time warpings of the second function. And just like Kendall did decades ago for rotation, now we can do this for functional data for time warping. So here is a quick example. I have a bunch of unimodal functions. If I just take the average as is, I get this average. And if I just automatically align it using this equation, very simple tool, it's a 10-line MATLAB code. We have packages for this. People use it in many different languages nowadays. It's become very popular. And it just do completely unassisted, completely automatic alignment. I do want to mention one little work, which is somewhat more recent. That's why I feel like it's a good idea to mention it, is how much alignment. Sometimes this framework is so good that it aligns too much, and I'll tell you what I mean by that. So I have a number of functions. This is a simulated data, a lot of functions, 50 functions. They are all somehow noisy versions of this red line, okay? And if I average them as is, as I mentioned earlier, the peaks and valleys are smoothed out because they are not lined up. They get smoothed out, which is the case here. But if I do it with full alignment, this program that I mentioned earlier, it aligns, it introduces small spurious peaks. It even aligns the noise. It's so good in alignment. It does too good of a job. So that begs the natural question is how much alignment is a good alignment? And so what we have developed is a framework, I'll mention it in the next slide, where we explore the strength of the penalty, and we try to see what is a good estimate for the lambda. It's sort of a balancing act between the two extremes. So let me mention it via an example. Well, so this is, okay, I'll work with the same example as before. This, as I mentioned, was completely non-aligned result, average as is. This is fully aligned. These are different intermediate alignments. What we do is we do all different alignments. So we explore this lambda spectrum from fully aligned to fully unaligned, and we measure, we count, we label the peaks in the intermediate results, and we see which peaks persist in this spectrum. As you're smoothing the data and averaging, some peaks die out, and sometimes even new peaks are created. And so we sort of look for these peaks, and we track, and we try to estimate this peak number 1 and peak number 3 persisted throughout the spectrum, so they must be the good ones. And so we find the lambda as the smallest lambda, which gives us this peak, and that gives us a result. Let me just explain it with some real data. So this was the hospitalization data that I had shown earlier. So if I take this data and I just take the average as is, as I mentioned earlier, we get 3 shallow peaks. I'm trying to find what is a common trend, common pattern across these 25 European countries in terms of their hospitalization curves. Okay? If I do this with full alignment, with our method, which we call fully elastic alignment, it sort of splits a peak into 2. Because of the shifting, COVID didn't occur synchronously across all countries. It arrived earlier, arrived later. The peaks and valleys were slightly shifted between different countries. And so that was the result of this full alignment. We studied these persistence diagrams. We looked at how the peaks persist, and we found there are 3 dominant peaks, and we stopped at certain lambda value. And our estimate for a common pattern was this green curve. It says there were 3 main peaks in this almost 2-year period, and they were quite dominant peak, not shallow, but quite dominant and well separated. Okay, and so this is an outcome of results of this type. So this was this discussion using scalar functions, just to give you some ideas about how to do registration, the notion of elasticity in comparing objects, and so on. Now I want to switch gears and start getting into more complicated objects. And this will go fast because there are too many details to bring it up at this stage, but I'll just show you some pictures and results. Suppose I want to study shapes of objects in images, planar objects. So I want to study shapes of 3D curves. It turns out this square root velocity function definition extends to curve in any dimension. You can study curves in a 1,000-dimensional space, exactly the same formula, and exactly the same registration problem. And one can do many different things with it. Just as an example, the pair that I wanted to compare, in Kendall's approach, you put some landmarks, and you compare them using these landmark representation. In our approach, you take the full curves, and you try to register which point goes to which point using this optimization. And then based on that optimization, you can start doing the deformations between the objects. Okay? These so-called geodesics. I just want to mention an example, an application, a case study that we did with this. This related to shape analysis of mitochondria in the slices of this skeletal muscle tissue in mice. So somebody took this electron micrographs, and by hand segmented these curves. Nowadays, one can do it with automated techniques. Deep learning networks can do this rather easily. But at that time, this is old data. Somebody did it by hand. They extracted the contours of these different mitochondria in the muscle tissue. And the questions they asked is that we have a certain number of factors. How do they influence the shape of mitochondria? So different kinds of mitochondria, of course, they have different shapes, but different cells in different parts of the body, do they have different shapes? The one very interesting example, I think that should appeal to all of us, was, does the lifestyle, so they have a population of mice, they were made to work, be on a treadmill all day long. Another set of mice, another population, just lying around, lazy, eating well. The lifestyle changes, does it affect? We know it affects the macrostructures in the body, exercising does, but does it even affect the subcellular structures, the shapes of mitochondria, on how we live our lifestyle? So this has been a long-standing, interesting problem, and we developed a 2-sample test based on this elastic shape analysis, and we showed that there is statistically significant difference between the 2 populations. based on the lifestyles, based on the exercise. For certain kind of mitochondria, it turns out we could prove it for SS mitochondria, not for the IMF mitochondria. And this test failed. We could not get the difference if you use the original techniques like the Kendall shape analysis. Moving on to more complicated objects, three-dimensional objects, surfaces, it turns out the notion of this sort of invariant Fisher-Rao metric extends even to more complicated objects. They are similar square root representations. And we have utilized these techniques to study body shapes, to study, I'll mention this particular project, where we looked at diagnosing PTSD. This is an ongoing project right now with Emory University and MD Anderson, where they look at subjects' MRI scans, and we extract structures of subcortical parts of human brain, hippocampus, putamen, and so on and so forth. Using this machinery that we have developed, we represent these shapes via some principal component analysis into some sort of a scalar Euclidean representation. We write down regression models using different cofactors, covariates, pull them together. And our goal is to be able to predict these so-called PTSD scores. And this was quite successful using these sort of shape analysis techniques. There was another interesting project was, what is a typical growth? What is an average growth in human being? You take some subcortical structure, so this is a hippocampus. In going from age 22 to age 90, how does one grow? So we looked across many databases, because usually databases are targeted towards a certain age group. So we looked at many databases, pooled the data across, and we tried to write down what is an average growth model over time from the age 22 to age 90 for certain structures in human brain. Continuing to even more complicated objects, these things we call tree-like objects. These objects are something which have a main branch and a few side branches, maybe even tertiary branches. Examples include neuron morphology and even botanical trees. The question was how to represent them, how to compare them. We think of these trees as a collection of curves. So curves I already talked about, but now if we have two trees, we have to figure out which part of the main branch matches with which part of the main branch, which side branch here matches with which side branch. So you can see the registration problem is getting more complicated. It's sort of hierarchical, but more complex problem. So we have developed mathematical tools. There are some nice algorithms that can be used for this registration. And at the end, we are able to deal with some really complex things. These are actual real trees. This is with collaboration, in collaboration with a group in Australia. They use 3D scanners to go around the tree and scan them and curate the data. A lot of painstaking work that goes into it. And so given these trees, we are able to compare them via these geodesics. These are our optimal deformations. We are given tree 1 and given tree 2. The rest are just simply deformations that our algorithm provides in trying to compare these two different trees. So here is another example where we took a very small plant and formed a geodesic between a small plant and a big tree in the hopes of using this kind of deformations as a model for biological growth, to be able to quantify what does it mean to be a normal growth. Okay. Oops, some pictures disappeared. That's fine. We have done a lot more work with this. We have developed statistical models. So in this database, we had lots of models of trees for one species. And for these using the techniques that I mentioned, we can compute what is an average shape. We can talk about means and covariances and principal component. We can even dare to put something like a Gaussian model. It's not going to be quite Gaussian in a classical sense, because these are somewhat nonlinear manifolds on which you are doing your analysis. But you can put some kind of a Gaussian type models, and you can generate some random trees. This was supposed to be a full set of random trees, but I'm glad at least you can see the last row here. But you can see we have a rich enough probability model to be able to generate new structures. These structures are not at all in the data set, in the training data set. These are completely new structures that are completely derived from the statistical model. So it shows you in some ways a validation or the richness of the model we have used. The last type of objects I'll mention is even more complex. These things are networks or graph. A lot of different people in this community and other communities are working on networks. So what is difficult about this is they are also collection of curves. So you have a bunch of nodes, and these nodes are connected by curves. And we are not only interested in which node connects with which node, these nodes are connected via curves which have shapes. So we are also interested in using the shapes of the curves which are forming these edges when we are trying to compare two networks. Okay, so this is retinal blood vessels. I don't know if you heard about it. Google announced maybe a couple of years ago that they will take pictures of the retina. They will extract these retinal blood vessel networks, and they will use this to predict an onset of cardiovascular event in your life 4 or 5 years in advance. They are so confident about using the structures of these things to even predict the bad things that are going to happen down the line. And so there is information in this network, but information is not only in just the connectivity, but also the shapes of these things. As we age, the morphology changes over time, and one wants to quantify it. One wants to talk about statistical analysis of this variability. So the way we do it is, as people do it in graphical analysis or network analysis, you represent a graph with an adjacency matrix, but the elements of this matrix are not just scalars or vectors. The elements of this metric is the shape of the curve that connects those two nodes. So it's a very high-dimensional, infinite-dimensional entries in this matrix. The difficulty in comparing two such networks is that if you look at, imagine two of these for two different subjects, and you're trying to compare, the problem is, again, registration. Which node in this graph will match with which node in this graph? And that turns out to be, on top of everything else we have discussed, another layer of difficulty, and that's called the permutation group. So I can fix the ordering of nodes in one graph, and I can try all possible reordering of nodes in the second graph to see how to minimize the distance between the two graphs. Just in the same spirit of Kendall, but now working with more and more complex types of registration tools. Okay. And so one can do that, and there's some simple examples. So we have, it's a part of the retinal blood vessel for subject one, for another subject, and we have ways of registering as many as we could. Not every node which is here is present here, and vice versa. So some edges die out, some edges are born, and the rest edges are deformed as much as you can in trying to compare these two things. So I will mention one last example in this. We are beginning to analyze, and we want to use these retinal blood vessel networks for predicting diagnosis or any health-related evaluation. So this is a simple example of moving in this direction. So we have this for 10 subjects, and we want to understand what are the common patterns and what are the variability. What makes them similar and what makes them different. We painstakingly picked up subjects as close to each other as possible because it's so complex a data that at the end, if we just take arbitrary data, we can't even visualize what we are looking for. So this was a very simple sort of hand-picked, cherry-picked example where we could compute an average structure for these 10 subjects, and this is overlaying of this average on top of these 10 structures. So you can see that there is a reasonable success in grabbing the macro structures out of these different individuals. And then one can, of course, try to do some sort of a covariance definition and do principal component analysis. And once you have principal component analysis, you can go down to some sort of a finite dimensional representation and bring the tools from multivariate statistics, regression models, and everything else that one wants to use for these. Okay? So the goal is, one last line on this, the goal is to use these shapes as predictors in the regression models to be able to predict measures of health indicators or other things that doctors are interested in. Okay, so I just want to stop here and make a quick summary of what my main message is. As data is getting more and more complex, in addition to statistics, of course statistics is central, but for these complex objects, it's very important to pay attention to the structures. Of course, the language of structures is geometry. So the tools from geometry and statistics go hand in hand in this kind of data analysis. it's a very important question to ask is which metric are we going to use to compute things? The example I gave you is functional data analysis. Perhaps 90%, perhaps 99% of the field uses the L2 metric for functional data analysis, and I showed you with some simple examples the limitation of that. So one has to actually think about what are the good metrics for comparing the structures that we are interested in. And so we have developed this field called elastic shape analysis. In some sense, it's built on the shoulders of these giants from way before, from decades ago. Perhaps they never thought about being able to use their tools in such modern statistical setups. But the key idea was to use Fisher-Rao type metrics, bring them back, make them more interesting, make them more applicable. And the key was this square root type transformation, which started with Anil Bhattacharya in 1947, is how to take different square roots so that complex metrics become simple, and we can now use the statistical tools which we have all learned and are comfortable with using. So I'll stop there. There are many, many applications, and this has been a wonderful journey. And I just want to stop there with all the people that I had mentioned. It was a fun experience, just sort of tracing the papers and the heritages and who was the student of who and how the ideas got passed down. So I'll stop here. Thank you very much for your attention, and I'll be happy to entertain questions. Thank you.
So thank you very much, Anuj. As I said before, if you can enter your questions via the app, and you can look at the questions on the app and support questions that you feel you would also like to hear the answers to, and I will go through one or two of them. So the first question I've got is perhaps a general question for someone wanting to get into this area. It says, the mathematics behind this method is scary. How do you advise someone wanting to get involved in this kind of analysis?
That's a good question. I get asked this a lot. My question is, one can learn it. It's like layers of onion. One can, for example, you can skip many slides in my talk, go straight to using square root representation, and in that space you are using the L2 metric, and that everything you have learned in multivariate statistics or traditional statistics class is applicable. So you don't have to go into deep about the origins of all the solutions that have been developed. So you can go straight to using the solution. That's one quick way of getting into it. Then of course you may be curious and say, why does this thing work? Why does this thing work so well? When we did alignment of functional data analysis, at first we were very happy that it actually did a good job. We did this then around 2010, and we are in 2023, and it's still the best tool for alignment of functional data analysis at the moment. And so then we went back and asked the question. In fact, now as I pointed out, it sometimes does too good a job of aligning functional. It's sometimes not, we don't want that much alignment. And so then we went back and tried to understand. So one can get started in this field by taking your data set, pulling out the right tools that have already been developed. There are computer codes out there. There are papers which are very applied type of papers in this field, and become comfortable with it. And I guarantee you that's no more difficult than any other field that you'll get started in statistics. Only when you start getting into the reasoning behind the metrics and the invariances and these properties, I assure you that will be very rewarding, but it doesn't have to be done on the first day. You can postpone that part. So.
Okay, we've got another question here. In regards to predicting tree growth, how does the method you've discussed compare with other methods, other algorithms that predict human aging?
So
human aging. I think the tree growth in terms of the retinal data.
Yes. Oh, sorry. I think I lost my slides. Yes. So what is the main difference between this family of solutions with a lot of other work people are doing in different fields? The single biggest difference is registration. Just think about the Kendall's approach and the elastic shape analysis. So Kendall's approach assumed that you get data, objects, which are pre-registered. If you go into the field of neuroimaging, look at all the statisticians, biostatisticians who are working in neuroimaging and asking questions about aging or predicting diseases, a vast majority of them assumes you get the data pre-processed in a way that is all nicely registered, that is all cleaned up and it's all set up for you. My claim is that in that pre-processing, you already lost important information. You have to be as close to the original data as possible as a statistician. That's such an important component that that's what we have to do. We have to not take the data, pre-register it, and then apply a statistical model. That registration has to be part of the regression because there might be information about the response variable y that you lost in pre-registering the data. And so that's the difference between our approach and some of the other approaches, is that usually when you do this anatomical study, the differences between population is being compared to differences within the population. That's always our goal. How different are the population versus how is the variable population within itself. The differences across are usually so subtle that these misregistration errors can inflate the variance within the population, and it reduces the separation of population. In anatomical, when you study brain structures and you try to predict diseases, the differences between populations are not glaring. They are very subtle. The doctors go through years and years of training to catch on to those subtle variations. And so we have to work as hard as we can to do the best alignment we can to be able to zoom in, hone in onto the actual differences that we are interested in. And that, in a nutshell, I'm not going to say that we have the best performances. We fail a lot. We fail far too much. But it's that registration error that we try to control as much as we can with this extra bit of work that we do. And I believe that's going to pay dividends down the line. Thank you.
So we actually have a question on registration. So does it commonly occur that there are multiple good possible registrations? And if so, have you looked at the impact of picking one particular optimal one?
Yeah, that's a very good question. One can ask this question in more complex scenarios where you're trying to register complex objects like the retinal blood vessels or trees. There's certainly multiple solutions. And so ultimately one has to rely on the domain specialist to say, is any of this make sense to you? So that would be the ultimate answer. But in a short term, as a statistician, what we can do is not sort of look for the best registration, but even think of it in a Bayesian sense. You can actually try to put some prior, think of it as a nuisance variable, and you try to integrate out this registration variability as a nuisance. And so if there are multiple peaks, multimodal solutions, you take all of that into account in some sense while you're integrating out the solution. So that's one way of doing it. Of course, there are philosophical opinions about whether one should go this way or that way. But it's a very important point. It's a very good question. There are always, how to align two very complex objects. There are multiple solutions. And, it relies completely on the knowledge of the domain experts to be able to select out the best in general.
So a question about another possible application. How could you use this work to study the development of urban areas and transport systems, including covariates like elevation, perm water?
Yeah, that's a good question, and I wish one of our proposals, which was just recently rejected, got funded. So it was on transportation systems. So there you have road networks, but the road networks are fixed. What is changing is the function along the network, the amount of traffic in different congestion times and different periods. And we were even bold enough to talk about comparing these sort of road networks across different cities in conjunction with people who are engineers in this field. So in principle, from a mathematical point of view, I think you think of these networks, and at each point on the network, you think of like a scalar function, which gives you the density of vehicle at that point. So one can think of it as functions on fixed networks and try to compare those things. That's the sort of technique that I didn't get to talk about here, but it follows rather easily. It follows rather naturally as an extension of what we have mentioned, and there is a potential, but since we haven't done that, I'm not going to go too far and say it's all doable, but we certainly are interested in it. Yeah. Thank you.
Okay, a question here. How difficult is it to compare images of differing quality?
That's also a very good question. I wish I was talking to you people more often. So for example, take two images with maybe a similar camera, but two different levels of blurring. Maybe in one image you are able to focus very well. In another case, you are not able to focus. It's sort of a blurred image. I worked on this problem in a project that involved face recognition. So you take pictures of people at different levels of blurring or clarity in the image, and you have to compare this picture. So in some sense, you not only want to be invariant to the rotation of the picture or the scale of the picture, like the zoom in and zoom out, you also want to be invariant to the blurring of the picture. Now, it turns out that blurring is yet another group action. It's a convolution by a Gaussian density, and one can talk about metrics that are invariant to blurring. So, I mean, there is some algebra involved. Maybe what I'm saying is not going to make complete sense to you, but this is yet another group. I sort of give a lot of credit to my PhD advisor, Ulf Grenander, not my PhD, my postdoc advisor, Ulf Grenander. He came up with this more generic framework where you have objects and lots of group actions. We already know about rotations and translations and time warping, but all kinds of, like if you change the light source in this room, if you move that bulb from there to there, my picture will change. The pixel values of my picture will change. But we want to be invariant to this change because it's still me. The identity of the person has not changed. So he has developed this very rich framework, which we call pattern theory, where you outline the groups you want to be invariant to. And you mod out, you remove this in an algebraic fashion by doing certain optimization with appropriate metrics. And so for different image qualities, for some things we have handle on, but not other things.
So a specific question here. Can you explain why L2 norm performs better than L1?
So firstly, I didn't say anything about L1 norm, and maybe I will not continue to say anything about L1 norm, but I'll say something about the L2 norm. And if you can take one drive home message, it's this equation. You know, why did not multivariate statistics just naturally extend to functional data? The hope was everything that we learned about multivariate statistics, we know how to analyze vectors, can we just take it to functional data? Here it's a Euclidean vector norm, here it's an L2 norm. There are so many parallels. There is a Parseval theorem that makes a fundamental connection. It was so tempting that L2 norm for functional data analysis would have been just perfect if it had completely worked. The simple reason it doesn't work, the L2 norm in a finite dimensional vector space, you do the rotation, it preserves the distance. If you do this kind of rotation, if you do this kind of time warping, this is, in my opinion, a one equation quantification of why L2 norm is not good for functional data analysis. I'm not saying it's not good for all kinds of functional data analysis. In the data analysis, we are interested in geometric structures. You're interested in peaks and valleys. I talked a lot about extracting common patterns, preserving peaks and valleys, geometric structures. You can always do something. You can always function. There is no law against it. But what are you getting out of that analysis? We have to pay attention to that. If your focus is on some interpretable results, then there are questions to be asked. And so in answer to the question, I didn't say anything about L1 metric, and I will not, but this metric versus this metric. That, in a nutshell, is the message of my talk.
So I've got one last question, I think, before we should break for refreshments. What would you advise to deal with overfitting, matching shapes that do not really match?
So I think I did build in one example for that, which was this situation. That we got the data. All of the data was generated by sort of corrupting this red line. We wanted to estimate the red line from this data. But if we just did our alignment, it over-aligned. It did overfitting. So there are many, many problem areas in statistics where we can overreach with our solutions. And so it's the same thing that we put a penalty, then of course the question is, how much penalty? If somebody else tells you that's good, you can go with that, but if you want to do this in an automated way, so that's why we have developed one solution for one specific context. There's always a danger of overreaching. But I will certainly take that versus underreaching. This is the current solution. If you use this data set and extract a common pattern, you get a blue line. That's the current state of the art. So I would rather take some other solutions over losing the structure in the data. So it's like to err on the side of adventure rather than tradition.
I will just ask one more question, because there's one question from someone who was mentioned in your talk. So I think that at least you deserve a question if you're mentioned in the talk. I think there are many tuning parameters. Any challenge in your applications?
So that's one tuning parameter, and case by case, we try to provide solution. I'm glad I had this slide up when this question is asked, because for this particular tuning parameter, we have a solution which has worked very well. In fact, this was a very satisfying result to extract a common pattern for the European countries in terms of COVID occurrences. If somebody asks you how many waves there were, in this case, it was, I mean, visually you can gather a lot, but automatically we were able to estimate the waves. One can actually relate these waves with different variants of the virus, Delta virus and Alpha, Beta, and so on. So this was very satisfying result. And so case by case tuning of parameters. One thing I do want to mention, in the original alignment method, there is no tuning parameter, none. Complete automated alignment, 15 years it has been existent, it is still the best method for aligning the data. We bring in the parameter to reduce the level of alignment, to do less work, to start penalizing. That's where it came up. So if you look at our work, where we try to use the Fisher-Rao metric, a Fisher-Rao type metric for doing registration, alignment, statistical analysis, statistical summaries, no parameter, none, fully automated. But of course, that's not always the best solution. That's why we bring in other things, and then we ask the question, how to tune those parameters.
So thank you very much, Anuj. Your talk generated lots of questions, not all of which we've had time to get to, and I'm sure Anuj will be happy to answer
Absolutely.
questions in the
refreshment.
refreshment.
I'll be happy to meet and
refreshment
send me
break.
a message. There is this beautiful software. I didn't know about this. But please send me a message. I'll be happy to meet people.
Okay, so it just remains to thank Anuj very much for his presentation.
Thank you. Thank you.